Hello, everyone. Welcome to Chapter 3, Part 3, our third lecture in this chapter. This will also wrap up Chapter 3. Today we're going to be looking at the organelles. So we will have talked about all of the major components of the cell after this lecture. Let's start with our not really attendance questions. One of them, the first one, I believe is actually from the previous lecture. I got the feeling that I did not answer this. I can't remember, so I'm going to go over this one again. Are fatty acids hydrophilic or hydrophobic? Next, what is the area between the plasma membrane and the nucleus called? And finally, what is the liquid portion of that substance called? So go ahead and pause the video and try to answer these. And let's go ahead and see what the answers are. Now again, I may have given the answer to this first one in the previous lecture. I can't remember, so I wanted to go over it again just to be safe. Fatty acids, they're lipids, and all lipids are hydrophobic. They are non-polar. So most of the cell membrane, the plasma membrane, is non-polar. Next, what is the area between the plasma membrane and the nucleus called? It's called the cytoplasm, and most of what we're going to be talking about today is going to involve the cytoplasm. Now, some sources list the nucleus as part of the cytoplasm as just another organelle. Some sources list it as its own structure. Our book lists the nucleus as not part of the cytoplasm, but as an organelle, so that's what we will follow. What is the liquid portion of the cytoplasm called? It's called the cytosol. So here again is our picture of just the generic cell. This is, I believe, figure 3.24 of your textbook. And this picture just kind of shows us a generalized cell with the plasma membrane here in yellow, and then all of the contents inside this big purple sphere is the nucleus. All of these other structures are the organelles, and the nucleus and the organelles are what we're going to be talking about today. But first, let's look at all of these fibrous structures that we see here. These collectively make something called the cytoskeleton. And just like your body has a skeleton, your cells have skeletons, but they are not made of bones, and they serve a few more purposes than what your skeleton serves. So there are a few different types of structures that make up the cytoskeleton. First, let's talk about microfilaments. Microfilaments are the smallest part of the cytoskeleton. And there's going to be some overlap between the different components of the cytoskeleton and what their jobs are. Each of them will contribute kind of to multiple jobs. We're just going to be talking about what the major functions of each part is. So microfilaments are involved with things like movement of the cell. So if the cell is moving from one place to another, microfilaments play a role in that. Also, microfilaments are involved in muscle contraction. So later in the semester when we talk about muscles, some of the proteins involved in that muscle contraction are microfilaments, something called actin. And also laying just beneath the surface of the plasma membrane, kind of this gray, hazy background material, is a mat of microfilaments called the terminal web. And the terminal web actually provides shape and support to the plasma membrane itself. All of those structures of the plasma membrane that we talked about in lecture 1 of chapter 3, they're kind of anchored to this terminal web. All right, I don't like the formatting of this, so I'm going to actually change that while we're lecturing here. There we go. Now, next, a little bit bigger than microfilaments are something called intermediate filaments. And intermediate filaments provide general shape of the cell itself, and it allows a cell to anchor itself to a neighboring cell. So here we can see some intermediate filaments in yellow, and then also in yellow kind of running across the cell. So these are a little bit stiffer than the microfilaments, so it allows the cell to kind of take shape around it. And then the largest parts of the cytoskeleton are these structures called microtubules, and that's these big kind of brown-orange structures here. Microtubules, they are the biggest part of the cytoskeleton. They give very stiff support to the cell. Organelles are actually anchored to the microtubules so that they can stay pretty much in place, but also the microtubules kind of form that railway or interstate system that I was talking about, so that if organelles, such as here we can see one of those vesicles that we saw previously, and here's that motor protein, well, other organelles also will have some motor proteins that they can actually move from place to place inside the cell along the microtubules. Also, microtubules are involved in cell division. So in chapter 4 and in lab, when we're learning about cell division, there's something called the mitotic spindle, and the mitotic spindle is made of microtubules. It allows certain parts of the cell to be moved during cell division, during mitosis. Now, off to one side of the cell, there's going to be kind of this hazy cloud called the centrosome, and inside the centrosome are two little structures called centrioles. So the centrosome is actually the structure that makes the microtubules. The microtubules sprout outward from the centrosome. So if we find a microtubule, we could trace it back, and we will find that it begins at the centrosome. Now, we will talk more about centrioles later in this chapter, but it is the centrosome that is responsible for making these microtubules. Okay, so now moving away from the cytoskeleton, and we will begin talking about the actual organelles of the cell. The first one we're going to talk about, again, as I mentioned, some sources do not list the nucleus as an organelle. They list it as its own structure. Most sources do list it as an organelle. I'm going to list it as an organelle, and your book lists it as an organelle. So the nucleus, as we mentioned when we were talking about DNA, the nucleus houses and protects the DNA of our cells. So our DNA is found within the nucleus. And the edge of the nucleus, this kind of purple area here, this is called the nuclear envelope. Sometimes it's called the nuclear membrane. Now, the nuclear envelope is a double layer membrane. It's a phospholipid bilayer, just like the cell membrane, the plasma membrane. But it folds back on itself to make a second phospholipid bilayer. So it's a phospholipid bilayer times two bilayers stacked on top of each other. So it's actually a 4-layer membrane in total, and that's just to give a little bit more protection to that DNA. It's very important to protect the DNA. Some things may need to get across that plasma membrane, that nuclear membrane. The nuclear membrane is really good at keeping things out or keeping the DNA and a couple of other things in. But if something does need to get in or out of the nucleus, it passes through these specialized channels called nuclear pores. Nuclear pores are very specialized channels in the nuclear envelope that allow very, very few substances in or out of the nucleus. Now, inside the nucleus, here we see this little dark guy right here called a nucleolus. there is always going to be at least one nucleolus inside of each nucleus. Sometimes there are more than one. When there are more than one, the plural are nucleoli. So one nucleolus, multiple nucleoli. And the nucleoli, it's their job to make structures called ribosomes. Now, ribosomes actually are made of two pieces that come together. So the nucleolus makes those subunits, those pieces of ribosomes. And then those pieces of ribosomes will leave the nucleus through these nuclear pores out into the rest of the cytoplasm, where then the two pieces of the ribosomes will come together to make a ribosome. So it's the function of the nucleolus to make ribosome subunits. And then those subunits leave and form actual full ribosomes out in the cytoplasm. So what are ribosomes? Ribosomes are the parts of the cell that make protein. Now, they will be very important when we get to chapter 4. We mentioned them in chapter 2 briefly when we were talking about proteins. What did we say proteins were made from? Proteins were made from amino acids. And ribosomes are made from proteins and rRNA, ribosomal RNA. The ribosomes, it's their job to take an amino acid and then take another amino acid and stick them together with a peptide bond, to take another amino acid, stick it to that, another amino acid, stick it to that, and just keep adding amino acid, amino acid, amino acid. So ribosomes take amino acids and build protein. Now let's talk about the other organelles. First, we're going to talk about something called the endoplasmic reticulum, or the ER. The endoplasmic reticulum, which we see right here, there's actually two different types. There's over here this kind of smooth looking type called the smooth ER, surprisingly. And over here, it's kind of studded. It has little pellets stuck to it. Well, those little pellets are ribosomes. Some ribosomes float around free in the cytoplasm. We call them free ribosomes. Some ribosomes are stuck to the endoplasmic reticulum. When that's the case, this endoplasmic reticulum is called the rough endoplasmic reticulum because it looks rough because of all the ribosomes. Now we can abbreviate this smooth ER or SER. We can abbreviate the rough as the RER or the rough ER. So you're welcome to abbreviate both of those, smooth ER or SER, rough ER or RER. Now, what are the functions of these? Well, first let's look at the shape. When we look at the shape, we can see one big difference is that there are ribosomes on the rough, but not on the smooth. That's going to provide a little bit of an idea of what the function of these guys are. Also, we can see that the smooth endoplasmic reticulum is a little bit skinnier, a little bit more frail looking, whereas the rough ER is a little bit thicker, a little bit broader, a little wider. Well, these open spaces, because the rough ER and the smooth ER both are hollow. They're like hollow tubes. And these spaces, the actual tubes of the endoplasmic reticulum, are called cisterns or cisternae. The cisterns or the cisternae are these branching web-like tubes. And that's in both the rough and the smooth. In the rough, the cisternae are a little broader. In the smooth, they're a little narrower. Well, let's look at the function of the smooth endoplasmic reticulum first, the one that does not have ribosomes on it. The smooth endoplasmic reticulum actually has 3 separate jobs. First, they are able to synthesize various lipids that the cell needs. So lipids are synthesized at the smooth endoplasmic reticulum. Also, the smooth endoplasmic reticulum plays a major role in detoxification. So toxins, or actually even other substances within our body that need to be broken down, the smooth endoplasmic reticulum does a lot of that. If we take recreational drugs, if we take actual prescribed pharmaceuticals, if we drink alcohol, those are 3 big ones that the smooth endoplasmic reticulum actually metabolizes, breaks down into less harmful substances. Now, it will do that with other substances too. Those are just the big 3 that we tend to use as examples. We find these in places like the liver and the kidneys, both of which are involved in a lot of detoxification. The smooth ER is in every cell of our body, but it's really, really extensive in the liver and in the kidneys. And finally, calcium storage. Now, calcium, previously we said was in a high concentration outside the cell, low concentration inside the cell. But let's go a little bit further, and this is going to be important a few times later in the semester, so maybe make a note of this. Not only is calcium low inside the cell, calcium should be non-existent inside the cell most of the time. The calcium that is inside of a cell should be stored in the smooth endoplasmic reticulum. And that's because calcium likes to attach to things. And typically, we don't want that to happen. Most of the time, we do not want calcium inside the cell because it will attach to things. And that can be dangerous. So if calcium is inside the cell, it's going to be stored in the smooth endoplasmic reticulum. Next, the rough endoplasmic reticulum. Well, the fact that it has ribosomes on it, and since we just talked about what ribosomes do, that should kind of give you a little bit of a hint as to what the function of the rough ER does. The rough endoplasmic reticulum synthesizes proteins, but not just any proteins. The ribosomes on the rough endoplasmic reticulum will build protein, and that protein will then travel into the interior of the rough endoplasmic reticulum, into these broad cisternae. And those proteins that are made on the rough endoplasmic reticulum are either going to be secreted from the cell through exocytosis, or they are going to become a part of the cell membrane itself. Remember all of those proteins that we saw in the cell membrane? They were made by ribosomes of the rough endoplasmic reticulum. Or finally, proteins that are inside of those modified vesicles that we mentioned in the previous lecture, which we will talk about today. So, the rough endoplasmic reticulum is going to make proteins or synthesize proteins that are either going to leave the cell, that are going to become a part of the cell membrane, or that are going to become a part of a modified vesicle. Next, the Golgi. And this is an organelle that actually will have several different names, and they all mean the same thing. The Golgi body, the Golgi complex, the Golgi apparatus. These are all the same thing, and just different books will give them different names. But know if you look at a different source and you see one of these words, it is this thing. So I just call it the Golgi. Now, it's the function of the Golgi to modify proteins that were made at the rough ER. So let's go back. The rough endoplasmic reticulum, this is where proteins are going to be made. The proteins that, like we said, are either going to leave the cell, become a part of the cell membrane, or become a modified vesicle. But when they're made here, they're actually not functional yet. When the rough ER makes a protein, it makes kind of a pre-protein. It's not functional at that point. So what happens is that protein we said was going to be fed into these cisternae. When that's finished, a piece of the rough ER breaks off and forms a vesicle that has that protein inside of it. And the vesicle then moves and joins with the Golgi. And now that protein is inside the Golgi. The Golgi is going to modify that protein. It's going to snip some pieces off. It may add some pieces to it. And then when that is finished, when the modification is done, a piece of the Golgi is going to break off and form a vesicle. And that vesicle now has the finished protein inside of it. So before we look to see the whole pathway, let's look at those vesicles that we keep talking about. We mentioned them in Chapter 3, Lecture 2. We said that they were essentially shipping containers. They were sacs that move around inside the cell, and there's going to be something contained inside of them, which is what we just saw. A vesicle made at the rough ER breaks off and travels to the Golgi. A vesicle made at the Golgi breaks off and travels somewhere else. Well, maybe that vesicle that broke off from the Golgi is one of these specialized vesicles that we see here. For example, there's something called a lysosome. A lysosome is kind of like the stomach of the cell. When we see LYS, that's a root word that means to break down or to split. And that's because here, this vesicle, the lysosome, the proteins inside of it are enzymes, specifically digestive enzymes. Now, when we go back and think about phagocytosis, for example, remember we only got to the point where the phagosome was formed? Well, that phagosome had a bacteria inside of it, or maybe it has some sort of debris inside of it, but we need to get rid of it. So a lysosome will come along and merge with the phagosome. And when a lysosome and a phagosome merge, it produces something called a phagolysosome. Well, what's inside of a phagolysosome? The bacteria or whatever the cell just ate, plus the digestive enzymes that were inside the lysosome. Put them together and what happens? That bacteria starts to be dissolved by those enzymes. Eventually, we've completely gotten rid of that bacteria or whatever else it was that the cell just ate. So lysosomes, yes, they're used for phagocytosis, but they're also used for anytime we have something in a cell, maybe an old organelle that's worn out and we need to get rid of it. Lysosomes can do that. Lysosomes can kind of engulf other organelles and break them down to be recycled. So lysosomes are kind of like the stomach of the cell, but they are just modified vesicles that contain digestive enzymes. Or maybe that vesicle that was made here, maybe instead of a lysosome, maybe it makes a peroxisome. A peroxisome is another type of modified vesicle. Peroxisomes contain enzymes, not digestive enzymes though. They contain a different type of enzyme. And they have this weird look under a microscope. They kind of look like they have a diamond or something inside of them. But peroxisomes contain enzymes that kind of behave like smooth endoplasmic reticulum. They can also detoxify substances, although really it's the smooth ER that's responsible for detoxification. But the peroxisomes can do that also. Peroxisomes can also break down lipids. But the really important thing that peroxisomes do is they're going to get rid of something called free radicals. Well, free radicals you've probably heard of, but may not know exactly what they are. Free radicals are unpaired electrons. Unpaired electrons. Electrons really like to bind with things, but unpaired electrons really, really like to bind with things. That's what free radicals are. Also, oxygen. Free radicals and oxygen. Well, everyone, when we think about oxygen, we think about, well, that's what we breathe for. It's very important. The truth of the matter is, there's only one single reaction in the body that we need oxygen for. And other than that one single reaction, oxygen is deadly. Oxygen will kill a person. Actually, every living thing has developed a way to get rid of oxygen when it goes somewhere that it shouldn't. Because oxygen also really likes to bind with things. Oxygen is an electron hog. It pulls electrons towards itself, and when it does that, it can create free radicals. So it's the job of a peroxisome to get rid of oxygen and to get rid of free radicals when these things show up where they shouldn't. So what does it do? It takes these oxygen molecule or these oxygen atoms and these free radicals, and it converts them to hydrogen peroxide. And then it takes that hydrogen peroxide and converts it to water. So it takes these oxygens and these free radicals and converts them ultimately to water, to something that's not so harmful. That's a peroxisome. So there are just plain vesicles, or sometimes they're called shipping vesicles. There are lysosomes. There are peroxisomes. But even lysosomes and peroxisomes are specialized vesicles. Where were they made? At the rough ER and modified by the Golgi. So here's going to be a little bit of a video about lysosomes. Again, this is attached to the PowerPoint, so if you can't hear it, just watch it on your PowerPoint. All right, so there we can see how lysosomes were made and what lysosomes do. But here is a picture from a different textbook that kind of sums everything that we just talked about up in one nice picture. And what we see up here at the top left, here is the rough endoplasmic reticulum. We've made some proteins on the rough endoplasmic reticulum. A piece of that rough endoplasmic reticulum breaks off. Here we see, again, pieces of it breaking off to form those shipping vesicles or transport vesicles. They travel forward with those incomplete proteins and merge with the Golgi. Now those proteins are inside the Golgi. The Golgi is modifying those proteins. And once they're modified and they are functional, a piece of the Golgi breaks off again as a vesicle. And we have those different pathways here. This is a protein destined to be secreted from the cell. So the vesicle travels forward, merges with the plasma membrane, actually becomes a part of the plasma membrane, and whatever was inside of it is dumped out of the cell through exocytosis. That's one way. Another option is the proteins travel forward and become a part of the cell membrane. So this is where all those proteins in the cell membrane came from. And lastly, here we have a vesicle, which could also be a peroxisome. If it was a peroxisome, it would go off and do its own thing. Here we see a lysosome merging with a phagosome to make a phagolysosome. Where did the phagosome come from? It was a piece of the plasma membrane that then came inward through phagocytosis. Now, before we move forward, I want to talk about something that I like to bring up in class because usually I get questions about it, and that is we keep breaking pieces off of the rough ER, or we break pieces off of the Golgi, and those pieces go and all of a sudden they're merging with the plasma membrane. Or over here we had a piece of the plasma membrane break off and merge with the vesicle. How does everything not just intermix, and why is that not a problem? Well, it's not a problem because in every one of these cases, the rough endoplasmic reticulum, also the smooth endoplasmic reticulum that we talked about, the Golgi, every type of vesicle, and a phagosome, and the plasma membrane itself. What are they made of? Every one of them is a phospholipid bilayer. So it's completely okay that a piece of one thing breaks off and becomes a piece of something else because they're all made out of the same stuff. Just pieces of the cell are moving around and becoming other parts of the cell. And that's completely okay because they're all phospholipid bilayer. Last organelle or major organelle that we want to talk about is the mitochondria. Now, I have kind of something to nitpick here with the textbook, and that is when I talk about this in class, I ask who here knows the role of the mitochondria, and like 95% of the class will raise their hand, and I will say it's not best thought of as the powerhouse of the cell. So who knows what the role of the mitochondria is? And of that group of students that have their hand up, another 80% probably will put their hands down because that's what you're taught in middle school and high school, and that's what sticks, and that's what you see in all of the memes, the powerhouse of the cell, which isn't wrong, but it doesn't tell you what it does. And the reason that I say I have a nitpick is because what does your book sum it up as? It says the mitochondria is the powerhouse of the cell. So yeah, it kind of is. That's one way of looking at it. But I want you to know what the mitochondria actually does. And the mitochondria perform something called cellular respiration. Now that just keeps popping up. We talked about it in chapter 2. We talked about it a little bit in chapter 3, part 1, I think. But we've talked about it a few times now. The mitochondria performs cellular respiration. What does cellular respiration do? It produces ATP. We saw the equation for it in chapter 2. So we're actually, when we do chapter 26 coming up, that's what chapter 26 is all about, cellular respiration. So don't worry about what cellular respiration is right now. We will get to it. But that's what the mitochondria does. It performs cellular respiration to make ATP. So it's making the energy that the cell uses, and that's where the meme powerhouse of the cell came from. Now, if we look at the mitochondria, it looks kind of weird. It's really neat, I think. It's got a membrane, a phospholipid bilayer, but it's another double membrane, kind of similar to the nucleus. It has an outer phospholipid bilayer and an inner phospholipid bilayer. And the inner membrane actually folds back and forth and makes these structures called cristae. Cristae are the folds of the inner membrane of the mitochondria. But all the way in the very, very middle of the mitochondria, inside the inner membrane area, there's this open space filled with this thick jelly-like liquid called the matrix. And the matrix is the innermost part of the mitochondria And there's going to be a lot of very special reactions that occur there that we will look at in chapter 26. Also floating around in the matrix is some DNA. And it seems kind of weird because when we look at it up close, it starts out looking like our DNA, but then when we look really close, it doesn't look like our DNA that much at all. Our DNA is in kind of these long strings, but mitochondrial DNA, or mtDNA, mitochondrial DNA is in rings. So if we looked at the mitochondrial DNA, it's in a ring instead of the long chain like our DNA. Well, these long chains of DNA, this is, animals, plants, things like that. But rings of DNA is actually what bacterial DNA look like. So bacterial DNA seems to be inside the mitochondria. Then if we look even closer and we start paying attention, the outer of these two membranes, the outer phospholipid bilayer, is the same phospholipid bilayer that we find in all of the other organelles that we just talked about. But the inner phospholipid bilayer does not look at all like the phospholipid bilayer in the rest of the cell. It actually looks like the phospholipid bilayer of a bacteria. So that really started to make scientists think, why does this thing that's kind of shaped like a bacteria, that has DNA like bacteria, that has an inner cell membrane like a bacteria, why does that exist inside of our cells? And as we were looking at them, they started doing something really weird. When we watch them, they go through cell division, but not when our cells are going through cell division. The mitochondria will just divide to make more mitochondria on their own. Or now and then, two mitochondria will merge to make one bigger mitochondria. So mitochondria will divide independent of when our cells divide. They seem to be like living their own life almost. Well, what we ended up discovering was that mitochondria at one point were bacteria. They were free-living bacteria related to something called cyanobacteria. But at some point, way in the distant past, they were phagocytized by another cell. But when that phagosome was formed, what we ended up with was mitochondria with the bacterial cell membrane trapped inside of a phagosome, which is, non-bacterial cell membrane. What do we have? We have something with two membranes, an inner membrane and an outer membrane. What usually happens? A lysosome would come along, merge, and we would break this down. But that didn't happen. Instead, this bacteria that we just ate is producing ATP, and the cell thinks, oh, well, the cell that just phagocytized it, the cell is getting free ATP from this bacteria that it just ate. Meanwhile, the bacteria is now protected. It doesn't have to defend itself because it's within a cell. So they develop this symbiotic relationship. The bacteria eventually becomes what is today a mitochondria. And the cell gets free ATP out of the deal. So the mitochondria is not a bacteria anymore. But historically, that's where they came from. All right, and now all living cells will have mitochondria in them, except for bacteria. Now here's our centrioles that we mentioned a little bit earlier. When we talk about centrosomes, centrosomes are the cloud, and there's two centrioles within that cloud, and the centrosomes, we said, build microtubules. But the centrioles that are inside that centrosome, kind of weird. We didn't really know what they did, so we did some experiments, and we took the centrioles out of the centrosome. And we watched to see, okay, if we take these out, let's watch to see what stops happening. That way we'll know what their job is. Well, the weird thing was, nothing changed. It appears as though the centrioles inside the centrosome don't do anything at all, because when we remove them, nothing changes. But we do find some centrioles elsewhere. When they're in the centrosome, there's two of them and they don't have a job. But there is a single centriole at the base of the flagellum in the sperm cells, or at the base of cilia at various cells in our body. So there they kind of form an anchor and a bit of a motor when we're looking at the cilia and flagella. So centrioles, depending on where they are, they either serve as an anchor and a motor for cilium and flagellum, or inside of a centrosome, they don't really do anything. Here is kind of a summary of the appearance and the function of all the organelles in a cell. And that is it for chapter 3. There will be another lecture on chapter 4 coming up. Then we will jump ahead to chapter 26. So chapter 3, chapter 4, and chapter 26 all together kind of form what I like to call the cell biology section of our class. After we finish those chapters, then we're going to cover chapter 5, which is going to focus on tissues, and that will wrap up unit 2. Okay, so hopefully everyone enjoyed the cell bio chapter here, and you're ready for the next couple. If you have any questions at all, please let me know. All right, take care.